Now let's get started the Unit 1 and Unit 2. Unit 1 is, I'm going to make a recording of this and then share it. I already went over the syllabus, so it's time to get started with the Unit 1 annotation and gene structure. So if you have any questions, yeah, please stop me and ask me anytime. So Unit 1 is about annotation and the gene structure. Why don't I actually get started to explain what I mean by annotation and gene structure. So before we do that, I want to make sure that you guys can access BFX3 server. How many of you have experience of using this server? Please raise your hand. Okay. Yeah, then the first, why do I need to access this server for this course? Because many software programs in bioinformatics is working in the Linux. And in this server, it's already pre-installed. You don't have to go through this installation process. So all you need to do is just get the access to it, to use it. Otherwise, you have to do the system admin work yourself, which is time consuming. So why don't I actually have some time so that to make sure you guys can access it. So let me, please try to access the BFX3 server. The IP address is the bfx3.aap.jhu.edu. Yeah, here is the BFX3 server location. And let me try with this computer whether I can do this. So I hope that this computer is connected to a VPN. And let me use and you can use anything, but let me try to use a PuTTY. Yeah. I'm using the Mac here. Mac has the terminal like this. So in that case, just one moment. SSH is a secure shell login, the command to get logged in, and use your own ID. In my case, as Yun16, bfx3.jhu.edu, aap.jhu.edu. And then use your password. Then I'm logged in. If I type who am I in one word, it gives me my ID. So then I'm ready to use any pre-installed genomic software. So let me stop here and then making sure that all the people can do that. If you can use the VPN.
But it's longer.
Yeah, yeah. But then, IP address is wrong. The IP address is. So this is the command. Any questions? Yeah, I want to make sure that everyone can log in. Oh, yes, it is asking, are you? I never heard that. Your JHU password. Just type it. Just type it like that. I know that, but actually type inside.
Oh, is it?
Yeah. The belief you don't type. Logged in. Can you take a look at my command? Who am I in one way? You're logged in. So everyone logged in, right? Okay. So there are we are going to use so many programs by getting into this server. You don't have to install, but if you refuse, I don't want to do that. Everything I want to download in my computer and install this. That's not impossible, but you will end up with spending huge amount of time doing the installation. Software installation is different from the Linux to the Windows or Mac. Software installation for Linux is a headache. So I want you to avoid as much as possible. Okay. Glad that I'm very glad to know that everyone now can log in. We may not use yeah. Okay, let me get started again. By the way, did you get the link for downloading this PowerPoint slide? I sent it to the chat. Zoom chat. Do you have the link? Right. Let me send it again in case you might not receive it. PPT for today. Is the link for the PPT. Okay. Now, another side note about the R. How many of you actually know how to use the R? Please raise your hand. Okay, probably half of us you are not familiar with R. R is a computer language, just like a Python, very versatile. But many software that you are using is actually written in R. So even though you may not program using the R, but it might be useful to use some software written in R, what we call R packages. So that's why I would like to give some brief introduction here. even though you are not required to program in R, but we are going to use some software within the R. To do that, you need to know how to start the R, right? So number one is you can use R in web browser by using Cloud R Studio. Now they changed the name to the project. So if you click that project.cloud, you click that. Sorry for that. Obviously, it's better that. Yeah, I'm not going to click. That once you click that, you guys have a Gmail? You can sign in with a Gmail. You don't have to create an account. You can sign in with a Gmail credential. So, yeah, why don't I actually show using my computer? The project.cloud is the website. And then here you can log in. Do you see that here? And you can log in with Google. Use your Gmail. Now click the new project. Okay. Let me just use the. So this what you are looking at is the R console. Here you can do the calculation. If you want to use R for calculator, you can do 1 + 1. The answer is 2. You can do the 2 to the 56 like that. The R is like this. So if you want to use R, one method that I recommend is using the project.cloud. Here, you don't have to install anything. Just use the R cloud. And is there any limitation? Yes, it's free, right? There is a limitation. The limitation is size of the memory, right? If you are doing the single cell RNA analysis, which requires big memory, it's not likely that you can do that here. Then you have to pay to get the what do you say, paid version, professional version or whatever. Then you can do that. So that's the NTT's model. But for most of our classwork, it will be fine with this. So by using this method, you don't have to download R and install R in your data. So I just went over to this I just went over this web browser. And so I told you that one of the issues is the random access memory. If you need a large RAM, what is and if you don't want to pay, then what is the alternative? Either you use your own computer or you use the server. If you have a big enough memory for your own computer, that's good. If not, you may want to use the server. And that's one of the reasons why I asked you to log in. So let me show that. So let me log in again. Okay, once you log in, I ask you to type the who am I to make sure that it gives you your login ID, your Johns Hopkins ID. But if you type capital R in the command line, no, this now let me step back. If you do the 1+1 here in the command line, you know what will happen? 1+1 is not a valid command, right? That's what will happen. It's not the command. But let's do the R, capital R, and press the enter key. Now you are in the R. If you do the 1+1 here, its answer is 2. So this is how you use the R in the cloud. The pro is now you have access to large RAM. But what is a con? There is no fancy graphical user interface, GUI, or the what we call the integrated development environment, IDE. So it's just a very like a manual. Yeah, question?
For the more intensive harnessing for this class, how much memory do you need?
I think you can do it in that R cloud. Yeah. Unless you move on to the single cell level. And to quit a queue, open parentheses and close parentheses. Do you want to save the image, what you have done? No, I don't want to save it. Now I came back to the the command line environment. Right. So yes, second method is using the VFX server. By typing R, you can actually use the R. The last method that I would like to say is you can install yourself in your own computer. Here is a website, the meter site. You can download the software R. And then you also want, after installing the R, you want to install the R Studio too. So it's a basically two-step process. But by using this web server, you can avoid those kind of things, installation problem. So I recommend number 1 method, not the 2 or 3. Okay. Any questions so far? So, okay. Now, let's get started about the content. Have you ever heard that GenBank? Genetic sequence banking? Like it's a database. Hey, here is some organism, and then I sequence it, and then where to record it. Yeah, GenBank. So GenBank contains many sequences.
Okay.
So if you use the buccal swab in your mouth, and you often find the Streptococcus pyogenes, that is a very common one. One question to you guys. If I sequence, is that sequence the same or slightly between my buccal swab and your buccal swab? Yeah, it's most likely slightly different. Then in the GenBank, a repository, does it host a representative genome of that Streptococcus pyogenes,
or you can submit your own. You can submit your own. So even for one organism, you can find many slightly different sequences in GenBank. Right?
So, I will give you about 5 minutes, and
Can you guys
go to GenBank, and then search there are so many records. A GenBank record of the AS Streptococcus pyogenes.
So what you click may be different from what your classmate clicks. And try to get understood yourself of what you did. What you will see there is annotation information of that organism. So I want you to read annotation information of your choice of Streptococcus pyogenes.
And then
I would like to ask one of you to volunteer to share your screen. So that we can go over that information together.
Yeah, feel free to share your screen. Thank you for sharing the screen. So, who is sharing the screen? Yeah, what's your name? Migno. Thank you, Migno, for sharing the screen. And Migno found a record of a Streptococcus pyogenes. There are many sequences of pyogenes. Okay. The first one says about how many base pairs of this sequence. It's 1,895,017 base pair level. And can we check out the sequence? Which one should I click to see the actual sequence of this record? GenBank is in GenBank, you upload the sequence, nucleotide sequence of that strain or organism at this sequence. So there should be a link to see the nucleotide sequence. Where to click it?
Faster sequence.
Faster. Yeah, can you click that? Yeah. The FASTA is a format that describe or convey the sequence information. FASTA file is composed of some information plus nucleotide title sequence. Nucleotide sequence starts from the line number 2. Then what about the line number 1? Line number 1 give us some general like ID. It always starts with greater than sign, followed by some comment or what we call ID.
So here the ID is AE009949.1, followed by the name of
this
streptococcus
biogen, particularly
MG
AS.410.635.01.FA23 and we are looking at the complete genome of that particular organism, of that strain. And this is an ACGT nucleotide sequence of it. Any questions? So after sequencing it in the GenBank, which is a database, you want to submit not only the sequence but also some other information. Can you go back to the previous slide? Previous page. So here, who found this and who submitted? Is there any paper to back it up? And authors and the journals and the comments and a lot of information. Can you go down? Yeah, related other sequences. Yeah, can you go down? Here, features. Yeah, the strain name is the MGA S8232, right? And then it has DNA gene. Can you highlight the DNA? The gene, yeah, that one. But then the gene location starts from 202 to the second base there, all the way to the 1557. So this is the DNA sequence is DNA sequence, and this, like I said, subsequence codes for the gene, which will make a protein product, right? And particularly the coding sequence is CDS stands for coding sequence. Yeah, 202 to the 1557. And then when you translate into 3 nucleotides starting from the 202, the 20324 code for ATG is the methionine. And can you highlight the methionine in the translation? M in the middle, yeah, go down, yeah, that one. Yeah, that methionine is the first amino acid. That is coded from position 2 to 3 and 4. So like that, you have the protein sequence of the gene. So, DNA is long, right? And then there is one gene from 202 to the 1557. It is the only protein product in this genome. No, there is a second one. Do you see that? Second gene is 1709 to the 2000, yeah, 845 DNA N. Likewise, you have multiple genes, right? Yep. So this genome or DNA is fully annotated. We know in this long DNA stretch of DNA, which part code for first protein, second protein, third protein, fourth protein. And we even know amino acid sequence of their proteins. So you just get the genomic record. Right? Any questions about the genomic?
Yeah,
Thank
thank you,
you,
Meghana, for sharing the screen. Let me share my screen
screen
again.
again.
So,
You can sequence the DNA by using single sequencing or NGS. So you can get the DNA ACGT thing. But that's not all. To be useful, you have to annotate where in that long stretch of DNA which part
code for the gene,
the protein,
and
and then
the details of that.
as you have seen. Then those are uploaded to the GenBank, and then other people do not have to invent the field. They just look at the record and then do the research on it. Yeah, right? Then, how you can find the protein coding region? Start and stop codon. Start codon is ATG. You have a three like a stop codon variant, right? Rebecca, can you share your screen again?
And
And
with the
FAST file,
FAST
FAST file.
Okay.
And then I want you to search ATG start code. Can you do the ATG? You saw that the first one is the start codon is 202, 200 second position. But that's not, look at that, how many ATGs are there? What's going on?
You also consider the stop for start and stop.
Yeah.
Stop is a encrypted.
Yeah.
So, not all of the ATG can be the real start codon locked up.
So
it's not
that simple. Hey, here's a DNA sequence. Let me
just
just try to find the ATG.
Hey, start codon.
No, it's not.
You need to actually see the pattern of this.
So that actually what's going on? Which one is the second? 200 a second.
ATG.
Pardon?
One line is how many? 60? So probably the first line around that. Yeah, fourth. Yeah,
Let's say that that's a the real.
Can
Can you
highlight
highlight about changing to a 50 base around the 50 base pair ahead of that ATG? Can you yeah, let's say so. Yeah, this region is called a promoter region, right? Promoter region is the region before the start code. What's happening? Transcription factors bind it to this promoter region, and RNA polymerase is recruited and start to transcribe to make messenger RNA. So, not all the ATG, but some specific ATG with that promoter binding motif can be the start code. So I'm checking at those what is that patterns of the sequence before ATG, we can say, hey, this is the real start codon location, or this is not the real start codon location. That is computational, like gene prediction. So again, if I summarize it again, finding ATG is pretty simple, but to identify the real start of the gene,
you have to find the motif of a promoter finding.
or in that promoter region.
Only those can be a gene, right? And you don't want to do this manually. You want to use the machine learning, or you want to use the software for doing that. So, yeah, thank you again for sharing
the screen.
So, the coding regions are particular segments of the DNA that code for the amino acid. You have seen it. Gene prediction is take a look at prediction. It's a prediction. You are doing computation. So it's kind of an automated way of annotating which regions could be the coding regions in a newly sequenced or partially sequenced genome. And then, in the GenBank, people usually, it's best to confirm it by experimentally, right? However, in many GenBank records, they just submit the computational prediction. Why? Because it's easy. You will learn how to do that in this Unit 1 and 2 and 3. Unit 2 is the gene prediction for prokaryotes. Unit 3 will be the gene prediction for the eukaryotes. Why do we distinguish between prokaryote and the eukaryote? Because the way how the different types of promoters are recognizing. So sequence patterns are different between prokaryotes and eukaryotes.
Question?
In the previous slide, like how you are explaining there needs to be a promoter for the start codon. So in case, how do we identify novel protein coding sequences which has a novel promoter motif?
The question is in case if you have a novel promoter motif, is this software can identify it? Probably not. That is the limitation. So that's why we call that gene prediction. And gene prediction is not perfect, especially when you are dealing with a novel promoter, because it's not trained on that. I think that just raised a good part. Gene prediction is anyway prediction. It's limited. And how can you actually confirm this? One popular method is by the RNA. Yeah, I will go over the detail in a few slides. So, the challenges in the gene prediction. Basically, the first challenge is it just to check the patterns of the sequence, and in biology, it's not, there is no 100%. Even though there is some slight variance in the sequence, promoters can still bind. So there is some room for error. And then, depending on whether you are studying eukaryotes versus prokaryotes, the mechanism of like a promoter sequence recognition is different. So you need a separate algorithm for eukaryotes. That's why Unit 2 is dedicated for prokaryote only, and Unit 3 is dedicated for the eukaryote. And then if you have a splice junction, then it's another complexity. right? Now, let's go to the gene annotation, the mRNA versus the genomic DNA. In bacteria, major difference between bacteria genomic DNA versus mRNA is the mRNA sequence does not have a promoter part, because for the mRNA to be made, promoter bind, and then it starts with the transcription start site. So those promoter regions are often missing, and then other regulatory regions. And one more thing to denote is bacterial mRNA sequence is polycitronic, meaning that, have you ever heard the intron, the operon? So one promoter and then multiple protein product, multiple genes. That's common in the bacteria, but not in eukaryotes. In eukaryote, usually one promoter followed by one protein product. So that's why you need a separate algorithm for the gene prediction for the bacteria versus eukaryotes. Basically, that is the content of Unit 1, but I want to touch on this discussion. So why don't we go to the canvas, and then here, and try to go to the discussion section of the canvas for Unit 1. I'm going to share my screen soon. Okay. So are you looking at this discussion in the Unit 1? And it says, please write the 3 discussion posts by Monday night. But you need to post your original writing on one of the topic that you choose this week by this Friday, the soft deadline, because you have to reply to one of your colleagues' original posts by the end of the unit, which is Monday night. And then in addition to that, you are required to the summary of your readings by the end of the unit. So here is the reading summary. So people usually prepare a PDF and then upload that PDF in this thread. So here is the reading summary, right? So reading is read 1, read 2, read 3, read 4. The ideal is you basically summarize all of them in your own language. Like could be like about a page or more, as you wish. So you just reply it and then attach the PDF. So that's how you get the 1 point for reading summary. Now, your original post. And let's say the pipeline annotation experience. Has anyone used any pipeline annotation tools? And if you have, please share experience. Now I can write my own like a thread. For example, I used a pipeline annotation software called something, and my experience is blah, 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 blah. And you write about it. And once you write about it, you post it, then your colleague, you post it by Friday, then your colleague can comment on that. So those are the 3 posts. Did I explain the way how you do the discussion? Okay. Any question about the discussions? Yeah.
For the reading summaries, are we required to submit 1 page for each reading, or all the 3 readings can encompass in 1 page?
It's your own discretion, but please summarize in the level that you are comfortable with. If you summarize more in detail with a specific example, that probably be more helpful for you in the future.
Okay. Should we initiate a thread for each of these the threads that are listed here?
No, you just choose
Sure.
only one,
Okay.
and then post your own like idea on it. That is the original writing on one of them.
Like thread 4 and 5 is written like compare results to others.
Yeah.
But if no one has posted, so how
Yeah, that's why by tomorrow night, Friday night, you have to post your original post to one of these. Then by probably on Saturday, you have a lot of posts that you can reply. So today is Thursday. Yeah. Okay. You saw that one gene, one promoter code service for the many genes, which is the operant example. That you try to go to the GenBank and then try to discuss about it in your own language. And that will be the original post. And wait for the Saturday, then people probably submit post many here. Then you have lots of things to choose just one and to reply to someone else's original post. Yeah, question?
So for us, this is Unit 1 and 2. Just Unit 1.
Unit 1 and 2 for, yeah. So now let me move on. So that is about the canvas. So far, I basically covered some like point for the take-home messages for Unit 1, saying that GenBank is, let me just summarize what we covered. GenBank is a database of nucleotide sequence. We usually upload not just the sequence, but gene annotation information. For that, we often need the gene prediction because we want to provide from here to here, it code for one gene, from here to here, another gene like that. And we use the gene prediction software for doing that. And then caveats are, it can be wrong because of some like a novel patterns or it's not 100% accurate. And then one more thing is, since the promoter patterns are very different between bacteria and eukaryotes, we need a separate program to do that. And we just covered the discussion. That's the end of the Unit 1. Do you have any question about the Unit 1?
the Unit 1 reading is in the modules.
Pardon?
The reading of the Unit 1.
Reading of Unit 1. You mean for the discussion post, right?
Mm-hmm.
So why don't I actually go over that here? So this is the canvas. For Unit 1, there are three part 1 and part 2 and part 3. So read this part 1, part 2, and part 3, and then open up your like a Microsoft Word and then summarize in your own language. If possible, insert some images, and then make a PDF and upload to that discussion.
That is a first discussion.
Where to upload? Here. By replying to the Module 1 reading summary.
Also, Professor, the deadline for reading summary is Monday.
Deadline is a Monday night.
Oh.
Yep. Okay. Yep. Now the time is 8:30. Why don't we take a 10-minute break? And then we will start at the 8:40.